Welcome to the Basics lecture on chromosomal inheritance. Several aspects of inheritance are linked to full chromosomes rather than just genes. Today we'll look at these topics. We started this unit by talking about single traits like flower color. An organism has two alleles of each gene that controls the phenotype. When an organism creates gametes by meiosis, only one of each homologous chromosome ends up in each gamete. This is called the law of segregation. Two alleles segregate into separate gametes. A hybrid cross of one trait is called a monohybrid cross. When Mendel was counting his pea plants, he realized that traits were inherited independently. Parent plants had genes for seed color and genes for seed shape. The F1 dihybrid contains all four alleles, and these four assort independently into the gametes. Some gametes get yellow and round alleles, and some are also yellow and wrinkled. This principle is called independent assortment. Genes on different chromosomes assort into gametes independently of each other. A cross between individuals heterozygous for two traits is called a dihybrid cross. Each plant has four possible gamete types. Stop the video and fill in the chart, and you can check Chapter 14 to make sure your answers are correct. Genes that are found on the X chromosome have a special effect on inheritance because they affect men and women differently. These are sex-linked genes. For instance, there are several genes that code for different versions of the opsin protein, a G protein-coupled receptor important for recognizing red or green light in the eye. If a chromosome has genes for both opsins, then the cones can distinguish both red and green light. But if the chromosome has lost the gene for either red or a green opsin, then the eye can't distinguish between them. Females have two X chromosomes, and as long as one of them contains both genes, they can make both proteins and have normal vision. Since males have only one X chromosome and one set of opsin alleles, they have a higher chance of exhibiting the phenotype of colorblindness. The X chromosome contains many more genes than the smaller Y chromosome, so almost all sex-linked traits are X-linked. Take a minute to read over this figure and its legend in Chapter 15 of your book and make sure you understand all three cases. Here's a question from the textbook. You can work on the answer and then check your results at the end of Chapter 15. Biologists and medical researchers often use pedigrees to track phenotypes and determine genotypes. Here's an overview of pedigree analysis. Squares and circles indicate males and females. Shading indicates the individual is affected and shows the phenotype. Relationships are indicated by lines. Here's the oldest generation, the next youngest generation, and the youngest generation. Pedigrees can help you determine whether a trait is dominant or recessive. In Pedigree 1, two affected individuals have an unaffected daughter. This indicates that the trait is dominant. If it were recessive, both parents would have to be homozygous and all their offspring would show the trait. In Pedigree 2, two unaffected individuals have an affected daughter. This indicates the trait is recessive. If it were dominant, one parent would have given it to their daughter, and that parent would demonstrate the trait. Females shut down one X chromosome in each cell as an embryo, but the X chromosome that's turned off is generally random and varies in each cell in the embryo. This occasionally produces a mixed phenotype in females, as different X chromosomes are producing different proteins in various cells. The classic example of this is pigment variation in female cats. If the active X chromosome has the gene for black fur, these cells will make black fur. If the active X chromosome in a cell instead is a gene for orange fur, those cells will make orange fur. More recent research has indicated that several mechanisms make the chromosome inactive. Here are two. The XI, or inactive chromosome, has more methylation of its DNA, and we know from Lecture 17 that more methylation means less transcription. Second, a gene called exist on the XI, or inactive chromosome, is turned on, which generates a large amount of untranscribed RNA, which coats the chromosome and prevents transcription. Lastly, let's look at recombination frequencies. This work was first done in fruit flies, so you need to be comfortable with how alleles are named in fruit fly genetics, which is different than pea plants. In this area of research, alleles are named when a mutant is found, so the alleles are named after the mutant, like black or vestigial. The normal allele is then listed with the mutant letter but a plus sign after it, indicating wild type, or normal, and this wild type is usually dominant to the mutant. You remember recombination from our discussion of meiosis. We've also talked about dihybrid crosses when we're interested in two different traits on the two different chromosomes. But what happens when the genes for both traits that you're interested in are on the same chromosome? Here we show a fly that's a dihybrid for two traits, body color and wing length, but the traits are on the same chromosome. In S phase, these chromosomes are replicated, and during meiosis, homologous chromosomes can cross over and change the alleles present on the same chromosome. After crossing over, some of the chromosomes are recombinant, meaning they've mixed the alleles that used to be on the same chromosome in the parent. Recombinants are more common if the two traits are far apart on the chromosome rather than close together, because it's easier to get a crossing over event between alleles when the genes are far apart. Before the days of DNA sequencing, scientists could use recombination to determine how close together genes were on the same chromosome. Close genes are considered linked, where linkage is defined as the tendency of two genes to be inherited together if they're close to each other on the chromosome. In order to measure how close two genes are on the chromosome, we can conduct a test cross. A dihybrid female is always crossed to a double mutant male because males don't undergo crossing over in fruit flies. Of the 4 possible offspring, 2 will be like their parents, but 2 will be recombinant. The proportion of recombinants is equivalent to how close together the genes are on the chromosomes. If there's more recombinants, the genes are far apart. Here's a question from the textbook on calculating recombination frequency. Work on the answer and then check your results at the end of Chapter 15. And this completes our basics video today. Thanks for watching.